Bismillahirrahmanirrahim. My name is Dr. Muhammad Latif and I am your instructor of the subject Principle of Animal Life 2. Today is the lecture number 13 in which we talk about gene mutation. What is mutations? Basically, the word mutation was similar to the French word mutation, which literally means process of changes. So the changes in the genetic makeup in the cell are called mutation. And the change in the genetic makeup means if there is a change in the genetic makeup, it will depict in genetic disorder in an organism. And one of the example of genetic disorder among humans is the sickle cell anemia, in which only one amino acid change and there is a disease which is called sickle cell anemia in hemoglobin, which is a oxygen carrying protein. What are the types of mutation? As I have told you that mutation are the change in the genetic material. So DNA is the deoxyribonucleic acid, which is the genetic material, and if there is a change in DNA, the whole genetic material will change and it has some consequences in the coming organisms. And you must familiar with the genes, because gene is a shorter segments of DNA which is responsible for the formation of a polypeptide or a protein. So if a gene is changed, it will produce a malfunction protein, and when a protein is changed, the original function of the protein cannot sustain and there is a malfunctioning protein in the organism which has some effects in an organism. And the last mutations are the chromosomal mutations. As I have discussed in my previous lecture in detail about the chromosomal mutations, because we all know there is a constant number of chromosome in an organisms, and if there is a change in chromosome numbers, there is a change in an organisms and it has some serious genetic abnormalities in the coming organism if there is a change. So what is point mutation? Point mutation is the change in the single base of the DNA. It's often known as single nucleotide polymorphism. And point mutation within a gene can be divided into two general categories. One is the base pair substitution and other one is the base pair insertions and deletions, or a frame shift mutation. We now in next slide we will discuss in detail what is base pair substitution and what is base pair insertion and deletion or frame shift mutation. What is base pair substitution? The replacement of one nucleotide and its partner from the complementary DNA strand with another pair of nucleotide is called a base pair substitution. And there may be following types of base pair substitution. The one is called missense mutation. What is missense mutation means? In this case, altered codons still code for amino acids and thus make sense, but this sense may not be the right sense. That's why it is called missense mutation. You must familiar about the codons. What is codon? It is a triplet code image. There is a 3 nucleotide sequence which pair a specific amino acid. So when a nucleotide in a codon is changed, it will lead to abnormal number of abnormal amino acids in a protein, and when there is abnormal amino acid in a protein, it can cause effect in the organisms. And what are the types of missense mutation? The type of missense mutation are harmless missense mutation. What is harmless missense mutation? In harmless missense mutation, there is redundancy of the genetic code, so some substitution mutations have no effect on the protein coded. In other words, a change in a base pair may transform one codon in another. The new codon is translated into the same amino acid. What does it mean? In an codon, there is a 3 nucleotide. The third nucleotide from the right side is none of the importance or a least importance. If there is a change in the last codon, sometime it cannot alter the amino acid. So this mutation do not have any effect on the protein or on the mRNA. And so when mRNA do not change, it will code for the same amino acids and the resultant protein are the same. So that's why this kind of mutation are called harmless missense mutation, in which the amino acid due to the change of codon does not change. And the other category of the missense mutation are the lethal missense mutation, in which the alteration of a single amino acid in important area of protein is called a lethal missense mutation. This alteration significantly alter protein, and such mutations are dangerous because they create useless or less active proteins that impair cellular functions. And one of the example is the sickle cell anemia. I have told you then there is only one amino acid change and the structure of hemoglobin is changed, and it will produce less oxygen. So it will carry less oxygen in an organism, and it has some lethal effects in that organism. And the other one is the called nonsense mutation. What does nonsense means? That the alteration in change of codon of an amino acid to a stop codon are called nonsense mutation. I have told you in my topic of genetic code, in which I have told you that some of the codon are not code for any amino acids, so that's why that codon are called stop codon. And if there is a change in the nucleotide in which a different nucleotide is changed to a nucleotide of stop codon, so the protein number of amino acid in a protein can be changed. If in a case of insulin, the insulin is made up of 51 amino acid. So if there is a change in insulin carrying mRNA, the resultant protein will cut the can cut the insulin protein by 30 amino acids. So the resultant insulin is not workable. So this kind of change, in which the change of codon of an amino acid to a stop codon, are called nonsense mutation. In this case, a point mutation changes a code for an amino acid into a codon that signals termination. So it will terminate the protein by not making the original number of amino acids a protein have. And the other one type of mutations are called base pair insertions and deletion, or a frame shift mutations. What is frame shift? Frame shift means, you must familiar about the DNA makes mRNA and mRNA makes protein. So mRNA has a certain sequence of nucleotide, and if there is a change in the sequence of a nucleotide, the resultant change will create a different protein. So insertion and deletions, or additions or loss of one or more nucleotide pair in a gene, this is called base pair insertions and deletions, and these mutations have a more dangerous effect. The mRNA is read as a series of nucleotide triple during translation. Therefore, the insertion or deletion of nucleotides may alter the reading frame of the genetic message, and such mutations are called frame shift mutations. These mutations occur whenever the number of nucleotides inserted or deleted is not a multiple of 3. And we will shift into the other topic, which is called mutagenesis. It's basically the mutagenesis are in which there is an agent which cause mutation in DNA. So what is a mutagen? A mutagen is an agent of substance that can bring about a permanent alteration to the physical composition of a DNA gene such that the genetic message is changed. And the mutagen, anything that causes a mutation, a change in the DNA of a cell, and DNA changes caused by mutagens may harm cells and cause certain diseases such as cancer. And what are these mutagens are? This can be the X-rays and ultraviolet rays, and this can be chemical mutagens. And these changes include alterations in the base sequence of DNA and changes that alter the structure or number of chromosomes. What is DNA? Basically, DNA interacts with the environment, and sometimes that interaction can be detrimental to genetic information because ultraviolet light from the sun can induce mutations in your skin cells. And mutation caused by chemicals. Most of the oxidizing agents, commonly known as free radicals, are substances that alter their base pairing capacities. Dioxin intercalates between these pairs, disrupting the integrity of the DNA helix and predisposing that site to insertions or deletion. And there is another chemical which is called benzopyrene, a known carcinogenic and a component of cigarette smoke, has been demonstrated to induce lesions at guanine bases in the tumor suppressor gene. Basically, these mutagens, what the mutagens cause? ये mutagens basically करते क्या हैं? You must know that DNA, you will go to the basic molecule biology, in which DNA basically these are the molecules, and molecules are made of atoms, and atoms made up of electrons, proton and neutron. So if an ultraviolet rays or an X-rays exposed to the DNA structure, it sometimes loses the electron or sometimes excites the electron. So when electron excites, there is a change in the atoms, and when there is a change in atom, different atoms are formed, and so the genetic makeup of DNA are changed. And what does chemicals do? Because chemicals are also molecules, and when these chemicals interact with DNA molecule, they make new chemicals. So the formation of new chemicals can cause change in the DNA structure, and when a DNA structure change, there is a change in the genetic material, and these genetic material change can bring changes in the coming organisms. And now I'll show you the animation which will further enhance the concept of gene mutation. Now, what are the basic genetic diseases caused by gene mutation? Some of the well-known inherited genetic disorders include cystic fibrosis, which is caused due to deletion mutation; sickle cell anemia, which is caused due to point mutation, in which only one nucleotide codon change and there is a different amino acid change; and the other is Tay-Sachs disease, which is also due to point mutation. Color blindness, cancer, tumors, all of these disorders are caused by the mutation of a single gene. And most inherited genetic disorders are recessive, which means that a person must inherit 2 copies of the mutated gene to inherit a disorder. इसका मतलब क्या है कि अगर ये मोस्ट ऑफ द इनहेरिटेड जेनेटिक डिजीज जो होते हैं वो रिसेसिव होते हैं. रिसेसिव का मतलब ये है कि जब दोनों रिसेसिव एलील जो है एक जीन के अंदर आएंगे तो वो अपने आप को एक्सप्रेस करेगा. अगर सिंगल एलील आएगा तो अपने आप को एक्सप्रेस नहीं करेगा. तो इस वजह से जो कॉन्सेंजेनियस मैरिजेज होती हैं, यानी जो ऐसी मैरिजेज जो कज़न मैरिजेज कहलाती हैं, अगर कोई ऐसा ट्रेंड आता है, कोई चेंज आ रहा है, मतलब उनके अंदर कोई ऐसी जेनेटिक डिजीज पाई जाती है, तो अगर कज़न मैरिजेज ज़्यादा होंगी तो वो जेनेटिक डिजीज उनकी फैमिलीज के अंदर फर्दर जाती रहेंगी. इसलिए डिस्करेज किया जाता है कि क्लोज रिलेटिव्स के अंदर मैरिज नहीं होनी चाहिए, क्योंकि 2 जेनेटिक सिमिलर एडल्ट्स आर मोर लाइकली टू गिव अ चाइल्ड 2 कॉपीज ऑफ अ डिफेक्टिव जीन.